Professor Baatout takes us to the Moon, Mars and beyond. She talks to us about her research in Antarctica on the effects on the human body of space exploration and space radiation. Enjoy. Professor Baatout takes us to the Moon, Mars and beyond. She talks to us about her research in Antarctica on the effects on the human body of space exploration and space radiation. Enjoy.
Okay, dear student, it's a great pleasure and a great honor to be here speaking in front of you about a fascinating field, which is human space exploration. And I will try in the next hour to give you a flavor of what is needed in terms of research and technology development in order to bring us all to Mars, which is for the moment our final destination. So since 60 years, we know more and more about the impact of space conditions on astronauts. And from the very first orbit of Yuri Gagarin that we will in fact congratulate for the 60 years anniversary in a month's time on the 12th of April. Since 1961 up to today, the length and the duration of space missions has tremendously increased from an hour and 40 minutes to now about 6 months to 1 year, which is the typical duration that our astronauts are staying in space. So we know more and more about the impact on their health, and we try, in fact, to counteract this impact, because we will see that it's not that an easy life for their body that the astronauts have got to face during their space flight. We are also living in an extremely exciting period of time, because all the international space agencies have gathered and have decided commonly to go for the same goal, which is to go back to the moon and to there investigate and develop a lunar outpost, a moon village, and thereafter to even conquer and to go towards Mars and to develop there a station and an outpost. So this time is absolutely fascinating because it asks and it demands quite a lot of investigation, research and innovation and creativity in order to be able to fulfill this endeavor to the moon and to Mars. And this program has a name which is called the Deep Space Getaway. But there is quite a huge showstopper, which is provided by ionizing radiation. And we will be speaking a little bit more about that in a minute. So going back to the moon and going thereafter to Mars is not just a European initiative from our European Space Agency, but as I said, a universal initiative from NASA, from the American side, but also from the Russian side. And other countries are also joining this endeavor. I speak about countries like Japan, like Canada, like India, like the Emirate Arab and also like Israel, for example. So it's really a universal endeavor to go back to the moon and also to go to Mars. And this Deep Space Getaway, which is the name of the program, starts with the Artemis Program. And the Artemis Program is to put back astronauts on the moon. This is the first part of the Deep Space Getaway. It has been signed and it has been decided a few years ago under the Trump administration, and it will continue now under the Biden administration in NASA. But if you compare, and just as an information here, if you compare the difference in terms of budget that was allocated to go to the moon on the first time with the Apollo mission, and now to go back on the moon, the proportion is quite different, from 4% of the federal budget of the US for the Apollo program to now 0.5% of the budget. So it's quite a tremendous decrease of the budget for a very important endeavor. If you look at the level of Europe, at the European Space Agency, the European Space Agency is kind of divided into 4 cornerstones, and these 4 cornerstones are very clear to remember. The first one is low Earth orbit. The low Earth orbit research is what we perform on the International Space Station. The International Space Station is about 300, 400 kilometers in orbit around the Earth, and where a lot of research, amongst others, research to prepare astronauts for longer missions to the Moon and to Mars. Then the second cornerstone is humans beyond low Earth orbit. Then we have the Moon, and then we have Mars. So these are the four cornerstones of our European Space Agency. But in order to really prepare astronauts on all points of view that we will look in a few minutes' time, you need, of course, you need space analogues. And space analogues are places, locations, like in Antarctica, different types of infrastructure that simulate partly space conditions. And ESA is also really working using these space analogues in order to study more in detail the impact of different types of space conditions on the human body. Space analogues, as I mentioned, can be Antarctica, and we will see later on what we can conduct in terms of research in Antarctica on the human body in order to understand what is the impact of isolation, of confinement, of extreme stress. And I was very lucky myself to go to Antarctica 2 years ago and to spend a certain amount of time there on the Princess Elisabeth station. And I will tell you a little bit more about this adventure. But there are also other infrastructures that are available in order to simulate some space conditions. One is the parabolic flights in which people, volunteers, or even astronauts during their training are basically monitored in order to understand what is the impact of a short time of weightlessness on the body. So this is one platform that we currently use at the Belgian Nuclear Research Center in order to test different aspects related to space conditions. Then you have another platform, which is the bed rest platform. And the bed rest study is a platform in which, we will see that a little bit later, more in detail. The bed rest platform is a platform in which people are lying down for a certain number of weeks or months. And it simulates the impact of weightlessness. It simulates because the body fluid moves and shifts towards the heads of the body because the body is inclined of about 6% with the legs a little bit higher than the head. Then we have different platforms of irradiation that we are using all over Belgium and all over Europe, where we can irradiate different cell types of the human body and different cell cultures in order to understand basically what is the impact of radiation on these cells. And then, if you are very lucky, you can also test the astronauts themselves. And that's what we also do. We currently have an experiment of following up astronauts who have stayed at least 6 months on the International Station, and where we test the impact of the space conditions on the immune system. So quite a lot of nice platforms, and just to give you an example for the moment, right now we have an experiment that just came back from the International Space Station, and these are rotifers. These are small organisms that have spent a month and a half aboard the International Space Station, and these are very interesting for us because we are going to analyze what makes them so radiation resistant. What are the molecular mechanisms that these little organisms have developed that we could better use in order to prepare astronauts, if necessary, for longer exposure to radiation, to cosmic radiation, on their way to Mars and on the way back. So you can see that on board of the International Space Station, you have quite a lot of European experiments that are taking place. And I just listed a few. On the human side, human research, you see that we have currently the Immuno2 experiment, which is the experiment on the astronauts themselves, where we measure different immune parameters before and when they come back. We also have another experiment, which is the Dosis 3D, which is continuously on board of the International Space Station, and which is basically how to measure the doses of radiation continuously that the astronauts are exposed to. And we will see that it's quite complex and it requires a continuous monitoring of the radiation doses. And we have another 2 experiments, the Rotifers that just came back, and then another one which is called the BioRock experiment, which is the first experiments to prepare moon agriculture. So to prepare through the use of bacteria and to understand how bacteria can extract from moon rocks or moon analog rocks, how do they purify, extract different types of minerals in order to be used for fertilizing plants in the future. So these are the very first experiments for moon agriculture. What are the effects of space conditions on the human body? They are quite numerous. You have the effect at the impact at the sensory motor level, also at the level of the spine. Usually astronauts gain 2-3 centimeters of height. They are a bit longer because the spine extends it a little bit. The bones are accelerating their aging. So you have an accelerated aging process, an accelerated osteoporosis at the level of the bones of the astronauts. At the level of the heart and the cardiovascular system, you also have a decrease of vascular function, reduced oxygen intake that can lead to a poor performance of the physically demanding tasks. Then we know that we have also a muscle loss that can happen in astronauts. And then, of course, we have the radiation, which is a very important factor that we will speak about later on. There are also differences between females and male astronauts. So this is also very important that in the next future, in the next generation of astronauts, the European Space Agency has announced this week that they are recruiting new astronauts, and you have for that to apply between the 31st of March this year to between the 31st of March to the 28th of May. And they are recruiting about 4 to 6 astronauts and then 20 reserve astronauts. And they are really calling for females to apply. So if you feel like it, please do that because it's really important. And it's said that the first person that will go back on the moon will be a woman. So we hope that we will have a female astronaut and that it will be a European female astronaut. Let's hope for it. So there are differences between female and males and different aspects of their behavior. Space radiation is the #1 risk for long-term space exploration, so really beyond low Earth orbit, which means going to the moon or to Mars. But our body, of course, is composed of different organs, and each organ has got, in fact, a specific radiation sensitivity. Each of us has got already an individual radiation susceptibility, but our body has got parts which are much more radiation sensitive than others. For example, the bone marrow, for example, the blood cells are very sensitive to radiation, whilst the brain is a lot less sensitive to radiation. And we know also, if we look at not only at the European level, but at the universal level, there is a big working group called the Multilateral Human Research Panel for Exploration. And they meet and kind of evaluate what are the specific risks for human space exploration. And what do they tell us? They tell us and they rate the risks into green, yellow, and red. Green means that we have ways to mitigate. We can really not take this stress and this risk too much into account. Whilst red means that we don't have any way yet to mitigate properly these risks. And what you can see is that radiation has got a red mark, certainly for missions going to Mars. It's not such an issue for missions to the International Space Station. There you see that it's green. But the longer the mission, the deep space duration, the higher the risk for the human body. On Earth, we are very well cocooned. We are very well protected by our atmosphere and also by our magnetosphere. And they block both about 99.9% of space radiation. So space radiation doesn't really arrive on Earth, and on Earth there is really no harmful effect of radiation from space, external radiation. But here, if you look at the increase of risks, if we go to the Moon and to Mars, you can see that the risk and the dose is much higher and is really increasing logarithmically. And for example, you see the level 1 is about 1 millisievert of dose, which is an annual dose on Earth that we all receive on Earth from the external environment. But if we look on the ISS, on the International Space Station, we have a dose which is about 150 times higher. And then looking at a trip to the Moon, we increase of another factor of 2 or 3, so we are about 300 times the dose that we would have on Earth. And then a mission to Mars would lead to an increase of dose about 1,000 times. So it's not nothing, it's really a risk that we really have to take into account very seriously. Here I'm not going to bother too much with the dose and the precise dose, because in fact the dose vary very much depending on the mission, the type of mission, the destination. If we speak about deep space, we will have a higher dose of irradiation than on low Earth orbit, on the ISS, because there on the ISS we're still kind of slightly protected by our magnetosphere. And we have, it depends also on the composition of the vehicle, of the space vehicle. We will see that it can have an impact on the habitat design and also on the activity of the sun. So these are really important factors that will change the radiation dose to which the astronauts will be exposed to. So a factor of 165, around 150 times higher on board the International Space Station in comparison with what we receive on Earth. A factor 300 higher from Earth if we speak about a lunar mission, and then about 1,000 times to 1,300 times for a Mars journey. Here in low orbit, we have the Van Allen belts, which are composed of electrons, and they are trapped particles, where we still have the Van Allen belt in which we have the International Space Station. But if we go beyond low Earth orbit, we have then 3 types of radiation we really have to kind of find solutions. First, we have the solar wind, which is a continuous flux of radiation of low energy electrons and protons and alpha particles. It's a continuous flux, but it presents a low hazard. So basically with the spacesuit or in the space habitat, there is no real risk of this solar wind. However, when the solar burst, when you have a solar eruption, you have then a flux of solar particle events. This is not a continuous flux. It can happen. It's intermittent. It lasts for a day or 2. But it brings a high energy proton flux, and this is quite detrimental. So when that happens, it can happen on the ISS. When that happens, the astronauts really have to shelter in some of the parts of the ISS which is very well protected. And it's very hazardous, but it's intermittent. It lasts for a day or 2, and then it calms down, and these solar particle events are stopping. But what doesn't stop is the third part, is the galactic cosmic rays that comes from the galaxy. And these are high energy protons. They are also alpha particles, and they can really be very damageable, detrimental for the cells. They are heavy ions, and it's a continuous flux for which we really need to find solutions by shielding or pharmacological solution or genetic solutions in order to make sure that we don't take too much risk for these astronauts. So it's an hazardous and for which we need more research. What is also important to mention is that not only you have these 3 types of radiation I just mentioned, but as soon as they arrive inside the space habitat, this first generation of irradiation can induce a myriad, a high quantity of secondary particle by interacting with the code of the space habitat. And so it's very important when we think about the spacecraft material to take the material that doesn't induce this secondary particle production. Where I work at the Belgian Nuclear Research Center, we are specialized in 3 different aspects. First, the dosimetry, which is understanding the doses that astronauts are exposed to on the ISS, but also being able to extrapolate these doses to what the astronauts are going to face when they go to the moon and go to Mars. So there is some kind of modeling approach as well. And for that, we are very lucky to have radiation dosimeters on board of the ISS continuously for many years now. Then we have another group, which is the group of microbiology, and they are especially devoted their time and energy to develop life support systems in order to make sure that astronauts will be able basically to eat and to have enough oxygen through the use of bacteria, cyanobacteria, and plants. And so it's very important to be able to recycle every drop of water, of urine, and of fecal material, and to be able to use that to feed plants and to be able to use that for growing vegetables and producing oxygen. So they are really working on different aspects of life support system. And then we have the radiobiologists that are working on understanding the impact of radiation on every part of the body and on every type of tissue, and preparing different ways, countermeasures, we say in space jargon, countermeasure to make sure that astronauts are going to face this moon and Mars trip in the most and the least risky way. So for the moment, we have a lot of experiments taking place on the ISS, because the ISS is really the critical place to mitigate and to develop all kinds of countermeasures before we go to Mars, and to estimate also human health impact and also the performance risks. And for the moment, as I mentioned, we have our detectors, radiation detectors there. We are measuring the radiation sensitivity of astronauts, of European and Russian cosmonauts. And we also have our life support experiments with our BioRock experiment on moon agriculture and life support in order to make sure that astronauts will have some food and will have enough oxygen. Radiation monitoring is taking place on board the ISS, and this red box that you can see on this picture shows you some of our dosimeters that are there and located on the International Space Station to understand space radiation and how it penetrates inside the space station. And we have for that different types of detectors that are active and passive. So passive are the ones that come back on the Earth and then they are measured. And the active dosimeters are the ones that give live the dose of irradiation continuously. And so these passive detectors that we have sent are recording the total radiation dose and are changed every 6 months. So every 6 months, astronauts receive new dosimeters from ours, and all dosimeters are brought back to the Earth in order to be measured. And that gives really an idea of the complete picture of space radiation on board the International Space Station. And you see here some pictures of Frank de Winne, our Belgian astronaut, and all the red dots that you see on the picture shows where he located the various dosimeters in order to really understand inside the Columbus module. The Columbus module is the European module, scientific module, and to understand in all the parts of the European modules what are the doses actually that reach the inside of this space station. I mentioned to you that when we have a solar flare, solar particle event, it is a massive flux of radiation. The astronauts have got to shelter in 3 parts of the International Space Station, and you can see the 3 parts here shown in red. And one of the parts is the water container, because water is an extremely good protectant against and stopper of cosmic radiation. So usually they go there, or they go also in the sleep station, and the sleep station is really surrounded with polyethylene, and polyethylene is basically the plastic bottles that we know. It's a long chain of carbon with hydrogen on each side. So it contains really a lot of hydrogen, and that blocks very well also radiation, cosmic radiation. This is another example of the research that we are doing. I mentioned to you this life support system that we are developing. This is a European endeavor, and the program is called the Melissa Loop Program. And the idea is really to have 4 compartments of bacteria, and each of them being able to fulfill a step of recycling of urines, feces, and rest of vegetables that are produced on board of the ISS. So you have first compartment which is a waste, liquefaction of the feces. And then you have the carbon transformation, the nitrogen transformation, and then the food and the water and oxygen production. And we have been, for the first experiments that we have been sending on the ISS was in 2002, and since then, basically we had quite a lot of experiments that have flown. The first experiment was with Frank de Wint during his first mission in 2002. Very simple experiments that we made at that time of cultures of bacteria in agar plates, and it worked very well. And thereafter we developed really more and more complex culture system for bacteria, amongst other, in liquid system to be able to monitor live the behavior of these bacteria and the physiology and the activity. This is a nice picture of our BioRock experiment, which is, as I mentioned, the preliminary experiment for moon agriculture, where basically lunar basalt is used together with bacteria to understand how bacteria can extract and purify some minerals in order to be used thereafter for fertilizers of plants. And we also have a very nice project which is called the Space Bakery, and the Space Bakery is the futuristic way of preparing bread for going to Mars. And it's really a very nice agronomical project, which is understanding how bacteria and plant interact, how to make crops produce the highest amounts in order to make a proper bread on board going to Mars. So it's a fantastic project that we also have started. And then in terms of radiation biology, there are different aspects that we are covering and that we are studying. First, developing biomarkers of radiation sensitivity, understanding and developing personalized medicine for astronauts in order to give them the very best medicine, the best dose of pharmaca, but also the best program of radiation exposure, because everyone has got a different radiation sensitivity. Some astronauts might be very sensitive to radiation, some others might be much more resistant. And that will play a role, certainly, in the moon village, because a lot of extravehicular activities are foreseen. A lot of time the astronauts will go out of this moon village and will go sample the moon soil and the moon ground. Will do some expedition on the soil of the moon. So they will just have the space suit on them, and the most radiation sensitive astronauts might not be so geared and so good in terms of health to do that. So it's very important to develop programs of personalized medicine for astronauts. We also test different types of pharmaca, radiation protectant pharmaca, in order to give maybe preventively, before they go out of the moon village, before they start the expedition, that they take these radiopharmaca and these radioprotectants in order to be fully protected before they are exposed to a higher dose of radiation. We also study radiation on the brain. We'll see some pictures on the microbiome, on the cardiovascular risks, on the skin, and on the immune system. And I will show you some more pictures later on. We are also very lucky to participate to different types of space analogues in order to understand on a higher, bigger cohort of people different types of space conditions. We have ground simulated infrastructure where we can simulate with random positioning machine basically cells that are then continuously floating. So like astronauts are floating in space, we can simulate it on the level of our cells and then do different types of cellular studies, understanding the impact of microgravity on the cells. And we can also irradiate with different types of cosmic spectrum irradiation and even do the two together. We're also very lucky to get samples from the bed rest study, people who stay for a certain number of weeks and months, and then we study the impact of this prolonged simulated microgravity, and we can better develop countermeasures. Then we have this confinement and isolation study campaign in Concordia, which is the Antarctica base from the European Space Agency, but also on the Princess Elisabeth station that you see on this picture. And then we have the parabolic flights that we use also as test platform for our hardware, for example, when we want to develop a certain type of experiment. And then we also do some in-flight experiments on the ISS or on the Photon. Let's have a look at the different types of experiments. We have here astronauts that are monitored regularly with our tests on the immune system, how their immune system basically is reacting towards the space conditions, all through the space mission, so before, during, and after, in order to understand the specific radiation susceptibility of each astronaut. We also use this bed rest study, as I mentioned, where people and volunteers are selected and they are staying for a certain number of weeks and months with the head a little bit lower than the rest of the body. And that really simulates the body fluid shift to the head, and that really simulates very well microgravity, in fact. And we get samples from these volunteers very regularly and that we study. We also we have irradiation campaigns in March. We have two very big irradiation campaigns in Groningen and also at Ganil at the Grand Accelerator in Caen in France, where we will irradiate really thousands of cells and cell samples in order to better understand the impact of the different types of radiation in space. We're also fascinated and interested by the skin because the skin ages in an accelerated way also in space conditions. And the skin is, as you know, the largest organ of the body. It has also vital functions like a barrier function, also it acts as an immune defense. And there are quite a lot of incidents that have been reported by astronauts, like small injuries of the skin, dryness, itching, and also delayed wound healing. And so what we know happen in space on the skin is that there is a thinning of the epidermis of about 20% in space. Also an increase in the collagen to elastin ratio. We know that there is also a loss of elasticity of the dermis and a kind of an atrophy of the dermal matrix, and all these phenomena are really related to an accelerated skin aging process. So we have sent some skin cells to the Photon M3 mission for 2 weeks in culture in an unmanned mission, and we studied the impact of space conditions on the skin samples. And it brought really a lot of results in terms of damage, accelerated aging of these samples, in terms of inflammatory processes that really allows us to understand better how skin behaves in astronauts for long-term space mission. We're also very interested in the cardiovascular system because the cardiovascular system, especially the endothelium is the thin line of cells between the blood and the rest of the vessels, of the blood vessels. And it's one single line of cells which is very sensitive to space conditions. And if these cells are dying and that are not kind of repopulated fast enough, you can have hemorrhage because then there is no really barrier for the blood to really remain inside the blood vessels, and it can spread in the rest of the body. So endothelial cells and cardiovascular risk is very important to understand and to mitigate, to counteract for long-term missions. So we have sent some cardiovascular cells to space, to the International Space Station with SpaceX. And that really helped us also to understand what are the process of cell death and cell aging of these endothelial cells. We also study brain cells and how brain plasticity can change in space conditions through different types of models. So we work mainly on mouse brain cells that we place in culture, and we put them in a simulated microgravity. We irradiate them with cosmic simulated radiation, and we'll see how the plasticity of brain cells is changing, how much time it takes for the brain to kind of be able, and brain cells to be able to reach their functional effects and functional rate as normal on Earth. So we can better understand what is the impact of a long-term space mission to the brain. And we're also studying the gut microbiome. As you know, the microbiome is considered, and the gut in general is considered as our second brain. And we know that the microbiome changes really a lot during a space mission. And the longer the space mission will last, and the higher the impact on the microbiome. So it's important to understand how to protect this microbiome and what are the main and the most important bacteria that need to remain, and what are the bacteria that decrease in terms of concentration all through this space flight duration, and how can we mitigate this decrease with maybe fecal transplantation or probiotics, all kinds of measures that we are developing in that way. We organize animal models in space research, so we organize quite often different topical days and conferences, and I would like to invite you also on the 12th of April. We are celebrating the 60 years of human space flights. So if you are interested, have a look. There is really a lot of information on the net for that. For the moment, we just have back our Rotifers from the ISS. This is our second experiment of Rotifers. We had one last year and one this year. Rotifers are very interesting microorganisms because they can live and survive in a dry form, dehydrated, as well as in a hydrated form. And so the first experiments we did was in a dehydrated form. And we've done also a hydrated form experiment on board. And they are so resistant to radiation that basically we irradiated them with a massive dose before they went to space last month. And then they continue to grow on the International Space Station. And now they are back, and we are going to further explore what are the mechanisms that they have developed during these 6 weeks aboard the International Space Station. It's also very important to further develop 3D models and life-on-chip models. This is really crucial because for long-term missions to Mars, we really will need some regenerative medicine in order to think about replacing an organ if it becomes dysfunctional during the course of a long-term mission. So we have different organoids that we have developed. Brain organoids. We are developing now also heart organoids, and the next one will be a gut organoid that we would like to develop, and all that for future regenerative medicine in space. And on the moon, as I mentioned, why do we want to go back on the moon? Because basically it's an archive of our Earth's own history. There is no magnetosphere, no general atmosphere, so no protection against space radiation, but it's a very important springboard for testing instruments, testing the behavior of the body, of the astronaut's body. And the idea is to have this moon village very soon, in a few years' time, with more and more astronauts that will be going there. So this international moon village in which humans and robots would work together on mining operations, on astronomy, and other projects on the lunar surface. When we will have this moon village, we will have a lot of extravehicular activities. So when the astronauts are going outside the moon village and do some exploration activities, for that they need a spacesuit. So we are also working on on the spacesuit, on what kind of sensors do we need on the spacesuit. It will be a kind of a second skin, but with also certainly detector sensors for temperature of the body, for radiation, of course, for humidity, for heartbeat, but much more than that. Really a lot of different types of sensors will be needed. And what do we know from the Apollo program? We know that there were about 20 extravehicular activities that were conducted, but for the future lunar program, we speak about 75 lunar extravehicular activity per astronaut to be performed, in which they will just have the spacesuit as only protection for them. So it's really important to develop the best spacesuit for that. You need to have a good spacesuit. You need to have a material which is resistant to abrasion, because you have the lunar dust, which is extremely corrosive, which really attacks most of the material, which is very reactive, chemical dust, resistant towards UV, resistance towards temperature, the oxygen, and of a light weight. And we'll have also a great attention to be able to walk, because these spacesuits will need to be used about 70 times per astronaut, so it needs to be used on a very regular basis, and it needs to be quite flexible. So flexibility, elasticity of the spacesuit is really important. So different materials are being tested for the moment, and in which we are also adding some sensors. But then the next step is Mars. Mars with 1,000 times more irradiation than on Earth. Mars is the most similar planet to the Earth. And through that, we want to really understand whether life existed elsewhere in the universe, into the universe beyond Earth. It's really a very important question for humanity. And we know that it has been once full of water and also warmer. It has a small atmosphere and it's a potentially habitable environment. What do space radiation induce in terms of health risk? First, an increased risk of inducing cancer, so what we call radiation carcinogenesis. Space radiation exposure may increase cancer morbidity as well as cancer mortality risks in astronauts. It can also, space radiation can also induce acute and late central nervous system effects, but also can affect the cardiovascular system and can also induce a kind of acute radiation response or acute radiation syndrome. But for the moment on the ISS, because only moderate doses of radiation are encountered, this acute radiation syndrome is never observed in astronauts. But for long-term missions to the Moon and to Mars, this might be a higher risk. And space radiation really needs a lot of research, because galactic cosmic rays are quite different qualitatively and quantitatively from what we experience on Earth in terms of X-ray or gamma rays that we use in the clinics. These galactic cosmic rays are very densely ionizing rays along the particle track, so it means that they can induce very complex and focused damage on the DNA. They can cause a unique damage to biomolecules, to cells and to tissues, and they really have a distinct pattern of DNA damage and a profile of oxidative damage and repair than we know with our X-rays used in the medical field and gamma rays. So they have a distinct biological pattern and maybe distinct health risks that we need to understand better. For the moment, the European Space Agency is also developing a new roadmap, the latest roadmap and strategy for research, for gaps of knowledge that we still need to fill, where we really need to push research and technological boundaries. The last roadmap was from 2016, and the new roadmap will be in 2021. So last year was a tremendous exercise in which I was very happy to participate very actively in order to develop the new roadmap for the next 5 years for the European Space Agency. And just a few words about the fact that we are not all equal in terms of radiation sensitivity. As you can see, some people can be very sensitive to radiation through mainly the genetic mutations that they have acquired from their ancestors. And some people are maybe more lucky and can be very radiation resistant. So it's interesting and very important, not only for astronauts, but also for the clinics, for radiation therapy treatment of our patients to really understand the mechanism of this radiation sensitivity, to better adapt the protocols of radiation therapy, for example. Radiation sensitivity varies with age. The younger ones are much more sensitive to radiation than the older ones. So in terms of personalized medicine and selection of astronauts to go to the moon and to go to Mars, should we take very young people, or should we take maybe people who are, as seen here in these pictures, from 50 years old? They seem to have much less radiation risks than when they are 10 years old. So very important to look also at this aspect of age, where you see a difference and a decrease of sensitivity with age. Radiation sensitivity is different also between women and men. So women are usually more radiation sensitive to men, and so depending on the age, depending on the gender, you can have tremendous differences, and depending on the intrinsic radiation sensitivity. So there is a need for harmonization between all the international space agencies for what dose are we going to tolerate for our astronauts of tomorrow that will be going to the moon and Mars. And for the moment, there is no harmonization. Each international and each agency has got the threshold of those in a different way. And we can see here that NASA has got differences, makes differences between females and males, and depending on the age, whilst our European astronauts, there is no difference of risk, so everyone is taken as equal. Should we continue like that? Should we change our threshold and our tolerated threshold and risks, depending on the age and depending on the gender? This is a very important question that at the international level is being discussed currently. Important also to develop biomarkers for precision medicine in astronauts in order to prepare in advance what type of pharmaca and doses of pharmaca each astronaut will need, because it might be very different from one to another depending on the genetic background and the susceptibility, not only to radiation but to other impacts. There are different types of biomarkers. We classify them into biomarkers of exposure to a certain stress, biomarkers of susceptibility, so biomarkers of late effects, so after the exposure of a certain stress, later on, years after, late effect can happen, and biomarkers of persistent effects. So all these type of biomarkers are investigated right now, amongst others, by my lab. Here a picture of the type of biomarkers, because of course these are genetic biomarkers from all types of style and kinds, from RNA, DNA, and protein level. So just would like to finish now with some nice experiments that we are conducting. Here you see some growing blood vessels on board the ISS. We're very lucky to have had these experiments on the cardiovascular system with Tim Peake as our astronaut, British astronauts, European astronauts, and together with SpaceX. We have here brain cells that we have cultivated in our lab. On the right-hand side, top right-hand side, you see a random positioning machine that can really allow to simulate microgravity. And with this beautiful instrument, together with some cosmic radiation simulation, we can really understand the molecular mechanisms behind brain plasticity and brain adaptation in space conditions. The gut microbiome is also very important in order to understand how the diversity in the gut microbiome changes all through a space flight duration. So very important, because the microbiome plays an important role in many fundamental and vital aspects of our life. And how can we counteract that? This is a nice picture of our rotifers. And I would like just to finish with Antarctica, where I have been spending some time 2 years ago on the Princess Elisabeth station. Antarctica is considered as the highest fidelity real-life analog on Earth for future lunar and Martian habitats on many aspects in terms of environment, which is quite similar monotony, the very poor environment, so quite simple, quite kind of monotonous in a way, seems always the same, even if monotonous seems pejorative, and I don't like that word very much, but it's, and when you go to Antarctica, you don't feel that it's a monotonous landscape at all, but it's the word which is usually used in the space jargon. In terms of the mission to Antarctica is more or less the same type of duration as a mission to space, to the ISS. The space habitat, a mission to Antarctica is in a confined and isolated habitat, an Antarctica station, like a space station. And then the social situation is quite comparable, where you have a small cohort of people, a small crew, international crew, with everyone participating to the different tasks and to the different housekeeping tasks, for example, with a very high level of solidarity and flexibility between each other, and also the psychological profile of the people going to Antarctica and going to space is also people daring to push the boundaries a little bit further. So they kind of have some kind of resemblance in terms of the psychological range of personality. Here's some beautiful picture of two Antarctic stations. The Antarctic Station Concordia, which is the European one, European Space Agency one, where we run experiments since and continuously since 2010 in order to understand the impact of extreme environment on the health when people are staying there for almost 13 months in a row and they have to face the winter, the Antarctic winter, with winds that can blow up to 240 kilometers per hour, with temperature that can reach minus 90. So they can't go out of the station. They have to stay for 6 months in a really isolated and confined environment. And that's where you really see how difficult it can be for the brain, for the mind, to stay really healthy and stable. It's not such an easy-peasy mission. Beautiful picture here of the Princess Elisabeth station. It looks like a space station. Going there it was really wonderful. It's the only zero-emission station where you have where energy comes from the sun. 200 solar panels are producing the energy to be used inside the station. You have 9 windmills that are also producing energy, and where I was really happy to conduct experiments on understanding the impact of this extreme environment on the human health, on different aspects, on the immune behavior, on the gut microbiome, but also on the heart and on the psychological impact. How can we improve radiation resistance of astronauts? There are different ways, different strategies. First, by enhancing shielding. This is a physical way. But there are other ways, also developing biomarkers, understanding which type of dose, which maximal dose each astronaut can get, and without having too much of a risk. Decreasing the metabolism of the body is also a nice area of research, which, like hibernation, is also interesting, because if you decrease the metabolism of the human body, then you can increase the radiation resistance. Biobanking, regenerative medicine is also a very important line of research in order to recreate, if needed, a certain organ for the astronauts, or biobanking as much as we can, biobanking the bone marrow and take it on board, have enough artificial blood, having, for example, an artificial lens in case lens opacity takes place and cataract is taking place, because the eye is quite sensitive to the space conditions. And with fecal transplantation, all these kind of regenerative technologies are important to develop. Radioprotectants as well. Radioprotectants is also a line of research which is very important. And space has got an incredible inspirational benefit for the whole community, from the young ones to the older ones, and also for you, students. Space research is very positively perceived by the public. It really inspires all type of education, any age and any type of education. It really helps to recruit new forces in the field. It's bringing really positive communication, and it helps also in this COVID time to bring positive news We had the Perseverance that landed on Mars, and I was very happy to do a live for that, and it was a beautiful moment of having this rover on the Mars surface, and this brings really a lot of joy to everyone. So the conclusion of my talk is it's important to have a better risk assessment for deep space exploration. So more insights is needed into the possible interaction and effects of spaceflight stressors, and also to understand and reveal altered cellular functioning in the body in order to be ready for Mars. If you are interested, there are different ways of getting more information. You can read this beautiful book, To Reply, No Reply, by Peter Rakers. It's a book in Dutch. There will be 3 volumes. It's a novel. And in which I'm playing my own character. So I'm recruiting the Martianauts that will go to Mars, and I'm training them before they leave to Mars. Then, if you are interested, there is also a TEDx conference on the specific aspects of space radiation. And I was very lucky also to be Space Personality of the Year 2 years ago. Some references if you are interested for the ones who really want to go and dig into the scientific aspects. But let's never forget that space research is extremely important for better treatment and for our patients. Basically, every technology that we can develop, every new medicine that we can develop is really has got a potential impact and a very important impact on our patients in order to cure better their diseases. So with that, I would like to invite you, if you want, to come to the Belgian Nuclear Research Center in Mol. Just drop us an email, come to visit when the COVID is over, and that we can move again freely. And with that, I really would like to thank you very much for your attention. And don't forget to apply to become an astronaut between the 31st of March to the 28th of May. So thank you very much.